All right. Okay, my name's Renee Duckworth, and I'll be moderating this session, and I'm also the first speaker. So I'm going to be talking to you about some work that I've been doing with my graduate student, Matthew Moling, on predictable response to unpredictable selection, an example in a passerine bird. So predicting evolution should be easy. I'm sure that's the message you've all taken from this symposium so far. And I would say, at least in the short term, right, we have a really clear predictive framework from quantitative genetics. If you want to understand the change in phenotype, at least for one generation, all you need to know is the heritability of a trait and the strength of selection on it. And this has actually worked remarkably well, and we have animal breeding and lots of laboratory studies that can attest to this. However, these short-term dynamics don't always scale up to much longer scales. And so this is nicely illustrated, I think, in this study by Gingrich et al., where they show that evolutionary rates depend on the timescale of measurement. So up in the, top corner here, we have the fastest evolutionary rates in things like lab experiments. However, you see really slow evolutionary rates if you change the timescale of measurement to something like fossil lineages. And one interpretation of this is that over the longest time scales, selection is much more likely to fluctuate. And the outcome of fluctuating selection can be very hard to predict. It depends on the predictability and timescales of environmental variation. However, we can make some, I'm an optimist in general, so we can make some very general predictions, right? If fluctuating selection itself is consistent over the long term and is predictable, then we would expect adaptive plasticity to evolve. At sort of the other end of the spectrum, you might have a short-term extreme event, which is highly unpredictable, and you would see a short-term change, but eventually the system might come back to some stable state, might recover. But what I'm going to mostly talk about today is this third sort of example, where you have long-term repeated events. So these events occur over and over again from the organism's perspective, but from the organism's perspective, they're highly unpredictable. So what would we expect to happen there? Not adaptive plasticity. If they're not something there's a cue for in the environment, then they can't really evolve that. So instead, I suggest that we should see the retention of multiple strategies. And we think we see an example of this in the species that I study, when we look at breeding timing, a really key phenological trait. So this is Western bluebirds, species I've been studying for the last quarter of a century. Makes me feel kind of old. So what I'm showing you here is data from our 20 years of data from our main population. And you can see that breeding date varies hugely in this species. So this is a short-term or short distance migrant. They show up to the breeding sites in mid-March. The first nests are initiated in mid-April, but some of the pairs are actually not initiating their first nest until the beginning of June. And so we have this 50-day span in breeding dates for this species, and this is pretty phenomenal for a passerine species. A lot of these birds, especially the migratory birds, will initiate nests in a very synchronized way within a 2 to 3 week window. And so one of the things that you might wonder is, well, this is all the data from 20 years summarized into one figure. Maybe it's because with climate change, phenology is changing, and this is actually reflecting some of the variation that we see over time rather than just steady variation within this population. However, we can look at that, and what we see is that breeding date itself is not changing over time. So this is breeding date, and I have residuals here because we corrected these estimates for age structure in the population, because older birds tend to breed a few days earlier than younger birds. But the main point here is there's really no directional trend. There's a lot of fluctuation across years, but no directional change. And this isn't too surprising when we look at April temperatures, which is the main cue for breeding initiation in this species. We see that over almost 100 years, there is no change in April temperature in the sites where we're studying them. And so perhaps it's not surprising that breeding date is not changing. But then that brings us back to the question of what maintains such extensive variation in breeding timing. And so what we hypothesize is that it's maintained by fluctuating selection. And we think we know the mechanism that's driving those fluctuations. Every year in our populations in western Montana, we get cold snaps that last for 2 to 3 days, and depending on their intensity and timing, they can kill up to half the nests in the population. So this is absolutely the main source of nest mortality in our system. And so why might this lead to fluctuating selection? Well, cold snaps, we think, may be creating opposing selection on breeding date. And that's because in general in birds, it's typically better if you breed earlier. That's because if you breed earlier, you have greater resource availability and you have more time for additional nesting attempts. So over and over in a lot of different bird species, that's a pattern they've seen. There's selection for earlier breeding. However, cold snaps, if you breed too early, they can increase the risk of offspring mortality during these cold weather events. And so this is just showing what that means for a early breeding strategy versus a late breeding strategy. If you initiate your nest in mid-April, like some of our earliest breeders do, then you have these old, hungry nestlings right in the peak time where we get these cold snaps during mid-May to early June. And that stage of the nesting cycle is most vulnerable to experience or to mortality from these cold snaps. Whereas the birds that are initiating later, they're still incubating during that time, and they seem to be much better able to weather these cold snaps if they don't have to feed nestlings while they're caring for the nest. And so one of the things that's important to note, though, is that we have this, or that the cold snap intensity is highly variable across years. So some years we get snow, as I showed you in that one picture. That was from June 7th in 2009, I believe. In other years, we barely have any cold snap weather at all. And so if we have a really intense cold snap year, we would expect these first nests to not make it, and that should shift the selection on breeding date for later nests. However, if we have a very mild cold snap year, then we expect, once again, that background selection for earlier breeding to predominate. And so to test this, we measured selection on breeding date across 20 years in our main study population. These birds breed in nest boxes, and so we have full phenology data as well as reproductive success data across these years. And so we estimated selection across years, and so this is a selection gradient here on the y-axis across these years. And there are a couple of things I want you to note here. First, most of the estimates of selection are indeed negative, but we do have a few years that go where selection actually reverses and is positive. And even in the years where selection is negative, it seems to be fluctuating all over the place. So it looks fluctuating, that's great, but we want to formally test for that. So we used meta-analysis to not only estimate the overall mean selection across the study years, but to calculate this heterogeneity score, which tells us how much, after correcting for error variance, how much of this is due to true heterogeneity in those measures across years, and it's significant. So 42% of the variation in selection gradients is due to true changes in selection across years. So we can then take those corrected estimates and say, are these fluctuations due to cold snap intensity? And what we found is that selection essentially reverses in years with intense cold snaps. So these are corrected selection gradient estimates in relation to cold snap intensity. And we're using here a biologically relevant measure, which is basically the percent of offspring that died in the population specifically due to cold snaps within a year. And what you can see in years where cold snap intensity is high, selection essentially reverses and turns positive. And so that's the summary from our long-term data, and it seems to be supporting our hypothesis that we have this fluctuating selection due to these cold snaps. But we wanted to test this and test the mechanism for it by going out and manipulating selection in the wild, trying to recapitulate those across-year patterns within a single season. And so to do that, Matthew did all this work. So he designated half the nest, about 20 nests, to be control and half to be experimental, and then watched the weather reports assiduously. And when all the other techs were like, sweet, we can sleep in tomorrow, he had to get up early and go out and provision all those nests that were in the experimental group with mealworms. And none of these nests had mortality in them during the cold snap, whereas the control nests did. And what we found is that this experimental treatment reverts that experimental group back to this background selection for earlier breeding. These black dots in the solid line are our experimental group that received supplemental food and did not have nest mortality due to cold snaps, whereas the dotted line here, and you can see those nests that suffered from mortality during the cold snap, basically is slightly positive. So it did essentially recapitulate within a single season those across-year fluctuations that we'd already observed. And so we can now, so the main message here for this symposium is we now understand the mechanism driving the changes in selection that we see across years in this population that we think are retaining this huge diversity of breeding timing strategies in this species. And so we can ask, now that we know that mechanism, well, how do we expect cold snaps to change over time? And what does that mean for the evolution of breeding timing in this species? Well, we then went and, or I should say Matthew went and gathered all the climate data for the last approximately 100 years. And what we found was really interesting because we thought that climate change might actually be causing cold snaps to be worse, like, you know, not only is the climate warming, but you get like more variation and that sort of thing. But what we actually found is that cold snap frequency is declining. This is a significant decline. And the other thing that was really interesting, and I don't know if I can really explain it, but cold snaps at our sites are getting later and later. And we've already started to see this like anecdotally in the field. So this year we had a cold snap with 6 inches of snow at our highest elevation site, June 17th. So they do seem to be getting more and more into June. And what that should do is that should make it more, that should actually reinforce selection for earlier breeding eventually in this population. And so the main message here is that we've talked a lot in some of these different symposiums and sessions about how to help species adapt to climate change. And in this particular case, ironically, I think, climate change will help this species adapt by essentially removing the opposing selection in what we predict to happen in the future then, and we'll see, I guess, eventually if this actually does happen, but we predict that once these cold snaps disappear or become late enough, we should see a much quicker movement of these birds to a single earlier breeding timing, or at least a variable breeding timing earlier in the season. And that's it. Thank you.
Thank you so much. Thank you all for being here. So I'm a new assistant professor at Texas A&M University, and today I'm going to talk a little bit about my postdoc work that I did at University of Minnesota with Suzanne McGaugh, working in repeated evolution of these cave phenotypes in these cave fish, Astyanax mexicanus. So this title is a little bit deceiving. At the heart of this talk, I'm really going to be talking about using repeated instances of evolution, repeated phenotypic evolution, digging in to see what the underpinning molecular basis is and evolutionary mechanisms of repeated evolution is, and then we can kind of take a step back and say, what does that tell us about the predictability of the evolutionary process? So a central goal in biology is to be able to reliably and accurately predict the traits a given organism will have based off of their genomic sequence. We now have some great examples where phenotypes have been mapped to underlying genetic variation, in some cases down to specific variants within a specific gene. However, in non-model systems, a lot of this work has been done with morphological traits, which are supposed to pop up here. Let's see. I'm trying, and that's still not working. How did we get it to go forward? No, okay. Let's see. How did we get it to go forward last time? I was up at like slide 2.
Okay, let's see.
Okay, you're just using
It's just not changing here. Yeah.
Okay, that's
You
fine.
just have
I'll
to
just
yeah,
look at this.
okay.
That's fine. Okay. Okay, we'll go quick. All right, so a lot of the traits that we know that we have good genotype-phenotype connections for tend to be things related to morphology or color pattern. These tend to be easy to observe and quantify, and they tend to have a simple genetic architecture, so maybe there's a single gene or a single genetic locus underlying variation in a given phenotype of interest. But what we are less certain about, and what we have less good data for genotype-phenotype connections are complex traits, and this involves things like behavior, where they can be difficult to quantify. There can be a complex genetic basis, so maybe there's a lot of underlying genetic loci that are controlling a phenotype-like behavior that's complex. We know that things like the environment can have a large influence on complex traits like behavior. And then if you're dealing with behavior specifically, we need to worry about the brain, which is highly complex and heterogeneous, both morphologically and functionally. And so this is a big problem, it seems like, but I think that there is hope for people who study genotype-phenotype connections in complex traits like behavior, because we have all of these genomic resources now available to us in non-model systems. And I think that we're able to make some great strides forward within the current time. And so my lab uses repeated evolution to get at the predictability of evolution and ask some of these questions about how predictable is the evolutionary process. And so I think repeated evolution is a great way to get at this. And so we have nice examples of repeated evolution when we look at things like the repeated evolution of wings across bats, birds, and insects. So this is more looking at like deep evolutionary time. We see repeated evolution of this coat color change in birds and mammals. But I think where we can really get better insight into how similar selective pressures underlie similar changes in phenotype and how those are connected is when we look at closely related populations within species, like these sticklebacks that I'm showing here, where we have an ancestral marine population that repeatedly have adapted to freshwater systems. Okay. And so one of the systems that my lab uses to study repeated evolution are the Astyanax cavefish. So in cavefish, we have these ancestral surface fish, which are shown on the left there, that are in surface river habitats that look like that little photo on the left. And so these ancestral surface lineages have repeatedly found their ways into cave systems in northeastern Mexico. And they have experienced convergent evolution on the same suites of traits that we often call troglodytic traits. And so they're cave-adapted organisms. They have morphological changes like loss of pigment in eyes and enhanced non-visual sensory systems. They have metabolic changes, changes to blood sugar regulation, and increased fat stores. And so I'll back up and say caves are very extreme environments. They're very harsh. If you're a surface fish and find your way in there, you're going to end up as cavefish food. You're not going to do well. There's low dissolved oxygen. There's not a lot of food. And so these fish also have interesting behavioral changes like loss of aggression and loss of sleep. So they have really substantially reduced sleep, where a cavefish will sleep maybe 2 to 4 hours total in a 24-hour period. And the loss of sleep is thought to actually be adaptive because they're spending more time foraging and looking for these really limited food resources. They all seem to have diabetes, but they do just fine. So they have this strange insulin regulation that's happening. They're all insulin resistant, but they have these fat stores that help them when they don't have a lot of food resources. Okay, so super interesting system, and it's been studied for over 80 years. People have been trying to look at some of the genetic bases of these different complex traits, but we still sort of don't know the answer to what are the molecular and evolutionary mechanisms of repeated evolution, because this has happened twice independently. And so, as I mentioned, I did this work, some of this work that I'm presenting today in Suzanne McGaw's lab at University of Minnesota as a postdoc. And so we sequenced 257 whole genomes doing resequencing from 18 cave and 8 surface locations. And does this work? Okay, so there are 3 different cave regions in northeastern Mexico that contain these cave ecotypes of the Mexican tetras. Cave and surface fish are considered the same species. They're interfertile. And so surface fish moved up into northeastern Mexico twice in 2 different bouts. And then the first bout invaded these Guatemala caves, and another bout invaded these El Abra caves. And so we have two distinct cave populations coming from two distinct surface lineages. There's also Migos caves, but I'm not going to talk about those today because they seem to be the result of an admixture event between one lineage of surface fish and another lineage of cave fish. So they're sort of too complicated, and we will ignore them for now. And so here's sort of just an overview of what I just said. We've got lineage 1, which I'll put in purple here, surface fish that gave rise to lineage 1 cave fish, and then lineage 2 surface fish gave rise to lineage 2 cave fish. So two distinct evolutionary lineages. We did a bunch of phylogenomics work to show that there does indeed seem to be just two independent origins of the cave phenotype. Okay, and so what sort of genetic mechanisms can be driving these sorts of patterns? So when we think about this, we could have something like I'm showing on the left where we have an ancestral phenotype and then the derived phenotype, and these little arrows here are representing the underlying genetic change, and so maybe repeated evolution is happening through very similar genetic mechanisms, targeting the same genes repeatedly leading to the same phenotype in these populations. Or, like I'm showing on the right here, maybe we have different underlying genetic changes repeatedly leading to the same phenotype. Okay, and when we have a scenario like the one on the left, the simplest explanation might be that gene flow has occurred, so maybe these derived traits have only evolved once truly and then have spread to the other populations via gene flow. We could also easily imagine selection on standing genetic variation has happened repeatedly and sorted these adaptive alleles out from ancestral surface populations into the cave populations, for example. And if we have a scenario like the one on the right, we might expect that de novo mutations, so truly unique mutations, have popped up and evolved in each of these different populations, and so we're getting unique underlying genetic changes leading to the same phenotype repeatedly. Okay, and then one other thing to consider, if we see a scenario like the one on the left, this might tell us that there's a really large role for deterministic processes and for genetic constraints. Maybe there's structural or developmental constraints or a large role for pleiotropy here. And so understanding the underlying genetic mechanism can really tell us something about the fundamental evolutionary process. Okay. So what I did with all these cavefish genomes was I wanted to ask what are the regions of the genome that might be repeatedly under selection and involved in repeated evolution. And so first I took sort of the classic approach where we take a bunch of genomes and we look where are there repeatedly, where's there evidence of selective sweeps repeatedly across these two different cavefish lineages, and I asked where is there neutral evolution happening in the surface fish lineages, so just looking for overlapping sweeps in all of these selected populations. And I used, to find these selective sweeps, I used Diploseek, a program by Kern and Schreider, to find overlapping sweeps. This basically takes PopGen data and it generates a whole ton of selection metrics and uses a convolutional neural network approach to then go to the genome, and each window in the genome is classified as neutral or under a sweep, basically. Okay. I then used a different approach where we're scanning for sort of, it's not quite scanning for selection formally, but scanning for parallel changes in allele frequencies away in the derived populations, away from the ancestral populations. And so this was using a program, AFVAPAR, developed by Jim Whiting, and it was based off of some math by Dan Bolnick, looking at parallel and repeated evolution. And so with this, we could look at two different types of repeated evolution happening in, so we'll talk about this with the cavefish as an example. So with locus reuse, here we have surface fish ancestral population leading to these cavefish lineages, and then we have the second surface fish lineage and all of its closely related cavefish lineages. Same thing in this other little tree here. And so with locus reuse, what we have is we have the cavefish alleles are different from the surface fish alleles. So maybe this one gene, the alleles are unique in the cavefish compared to the surface fish, and allele frequencies are moving away from the surface fish in both cavefish lineages at this locus. So the same locus is being a target of repeated selection or repeated evolution, but it's unique alleles. Or we can have allele reuse, where we have this true example of the same exact allele rising in frequency in both cavefish populations, for example, and that is different from the surface fish allele. Okay. So we identified all of these potential candidate genes that are targets of repeated evolution using the overlapping sweeps and then the scan for repeated selection. And what we found is a lot of discordance. And so this sort of tells us that the approach that you use really matters when you're finding your candidate genes. And so the overlapping sweeps and then the locus and allele reuse did not really overlap much. And we think that that reason is because we had to be really stringent with our overlapping sweep data set and having only neutral evolution in our surface fish lineages, and we had to have a sweep in both cave fish lineages because of the next approach that we use, which I'll tell you about in a moment. And also this AFA-BAR program might have been a better choice because it considers phylogenetic context when it's looking for these selection scans as well. So that's all I'll say on that. And then just very quickly next, we looked at the underlying mechanism of repeated evolution once we found our candidate genes. We said, okay, these are our candidates for repeated evolution. What's the underlying mechanism that's repeatedly driving these to higher frequencies in the cave fish? We used distinguishing modes of convergence by Kristen Lee and Graham Coop, and we looked at whether each of these candidate genes for selection were evolving via independent mutations, standing genetic variation, or migration. And I'll just say we looked at this both in cave fish, so shared sweeps across cave fish lineages, and as sort of a control, we looked at shared sweeps across surface fish lineages, and here's the punchline here. So on the x-axis, I have the mode of convergence, so independent de novo mutations, migration, or standing genetic variation. And surface fish, which are in the blue, we saw that a lot of the repeated sweeps were due to selection on standing genetic variation, 95% of them. And in cave fish, standing genetic variation also plays a large role. But in cave fish, we also saw that 40% of those genes that were candidates for repeated evolution had de novo mutations in them. Okay, so that was interesting. And also notably, those genes that were repeatedly under selection tended to be longer compared to genes in the rest of the genome. And so this tells us that genes that are longer might be targets of selection and repeated evolution. And this pattern was really driven by de novo mutations. And so if you have a larger landing pad, there's more mutational opportunity, basically, and loss of function mutations happen a lot in cave fish. I'll just leave it at that. I was going to talk about OCA2, but I think that we're running out of time, so for the sake of time, I'll just say we have evidence, and we've used CRISPR. Our colleague Joanna Kowalko has done CRISPR on some of these really long genes that we know have unique mutations in different caves, and we've gotten phenotypes, and so that's all really exciting. And so just to sum up, we think that selection is happening via standing genetic variation and de novo mutations, and that larger genes tend to be the targets of selection in repeated evolution. Okay, we made it. Thank you.
Well, thanks everyone for being here. I'm not sure what that blue box is, but hopefully it'll go away in a second. All right, so I'm an associate professor at Case Western, and I'd like to introduce my other co-authors to you on this project. So Sarah Diamond, also at Case Western, she requested the Badass Hawk photo to be used. And Logan Kolaski, who's a new PhD student in my lab in his second year. And so this work is now kind of in press at ICB for most of the stuff I'm going to talk about. And you can check it out if you want through the DOI or the code. Okay, so to fit in, right, with the theme of our session here, of our symposium, I'm interested in to what degree is evolution predictable and repeatable to environmental change, right? So how repeatable is it across species? How predictable is it based on our understanding of questions we're asking? And in this case, how rapid is it? How well is evolution keeping pace with environmental change? So the environmental change that I'm interested in is something that I think a lot of us are. It's environmental warming. So both kind of global warming, as shown here, but also localized warming due to land use changes such as urbanization, right? And obviously this is a pervasive, selective pressure that all organisms are having to deal with at a fairly rapid pace. So how are they doing it? And so there's multiple ways, that we can, make predictions and test predictions with this question. And, so some, like historically, we can use kind of methods of phylogeography to ask, how have species evolved in response to past warming, right, climate change in general, or colds, and ask from that how they might respond in the future. These work well, but there's limitations. This is long-term evolution, and it's kind of hard to get process from pattern from this sometimes. You can also do really cool experiments like resurrection studies or external evolution, but there's only a few systems that are really good for looking at these kind of questions. So I'd like to advocate that we can use a natural experiment or an unnatural experiment as well to kind of test predictions about how a species will respond to warming, and using urbanization, right? So urbanization creates local heat islands, right, that warm up the environment in kind of ecologically relevant ways that are fast and occur over fairly short distances that populations will have to respond to. And you find a wide taxonomic diversity in urban heat islands that we can use to look at these questions. And it's already established experiment for us. We can just go out and look at the animals. Right? And so there are, people are taking advantage of this. There's more and more studies of looking at the thermal physiology within cities and looking at the phenotypic and evolutionary responses that we see. And so, what I'm doing here is we went and did a meta-analysis. We quantified these responses that we could find and looked at overall kind of patterns. So I'm kind of asking 3 specific questions here. Is there evidence of convergent evolution of thermal physiology and some associated traits I'm going to talk about across diverse lineages and urban heat islands? Are these predictable based on our basic understandings of thermal physiology from kind of first principles? And, is this keeping pace with the rate of warming in cities? And so for our meta-analysis, we went through and we found studies that compared urban and non-urban populations of the same species that measured critical thermal limits, so CT mins and CT maxes essentially. And so we found we have 22 species for heat tolerance with multiple kind of replicates within that. 7 species for cold tolerance, not as many people measure that. And then we also measured body size for those studies, right? Not on their own, there's plenty more for that, but we were interested in how body size interacted with thermal tolerances. And so this is composed of both genetic data from common garden experiments and also just phenotypic data from wild-caught animals just brought into the lab. And we have a pretty wide diversity in this database, in all ectotherms, but otherwise wide diversity. And so, what predictions can we make? So if we, just take the classic thermal performance curve, what we're pulling out from these studies is, the critical thermal minimum and maximum, so these physiological limits, right? We don't really have whole performance curves for most of these. And we might just predict, just a simple prediction that they'll be shifted, right? That you can shift the thermal limits, maybe the curve won't look the same, but they'll all go up. So a loss of cold tolerance and increase in heat tolerance in cities and in urban heat islands. And from this, we can make a bunch of more, more predictions, right? So first, these shifts in thermal tolerance, we think we're going to predict will be repeatable across all the species, most of the species we looked at, right? Broadly repeatable. There'll be greater heat tolerance in urban populations with a loss of cold tolerance as well, maybe because of relaxed selection or indirect selection through heat tolerance or other traits. And the larger the heat island effect, we expected more change, right? Greater evolutionary change in response to stronger selection. However, if you start off at a higher thermal capacity, higher heat tolerance, that might result in relaxed selection, or if you're already pretty high, maybe there's upper thermal limits, evolutionary limits, loss, lack of heritability or something that limits your response. Okay. And so let's take a broad look of first just overall, what do we find the mean effects? So we have heat tolerance on the left-hand side, cold tolerance on the right-hand side. So overall, you can see the dashed 0 line there. So they shifted as we expected broadly. There's a lot of variation here that we'll try to explain in a minute. But heat tolerance, right? Populations generally are evolving greater heat tolerance, and they're also losing their cold tolerance. This is true if you subset just the common garden genetic data or the phenotypic data overall. The phenotypic data is kind of biased upwards, probably because of plasticity, right? And let's go on and test these other predictions. So bigger heat islands, what happens? Well, broadly, you get greater responses, right? So the larger the difference between, and we pulled this data from studies when it was there. So what's the actual habitat temperatures that populations are experiencing? Or we grabbed them from the GPS coordinates and calculated them ourselves. And so they are, the bigger the heat island, the more effect. In the circles here, the circle size is just our model weights, right? But you see that red line there too, right? That's our 1-to-1 line. And that suggests that populations are not keeping pace with that rate of warming in cities, right? And we can take a look at that lag directly and model it, which we did on the right-hand side here. And you can see it's pretty substantial, right? So it comes out that it's like, 1.6 degrees Celsius mean lag. The difference between how much the population in the urban is responding to that urban warming compared to the magnitude of the urban heat islands. And it increases with increasing urban heat island effects, right? So the lag gets greater. If we look at that kind of baseline tolerance idea, so what's the baseline thermal tolerance of the rural populations? It does have an effect, and it's the way we predicted. So populations with higher baseline tolerances, they're paired populations in the city. evolve less, right? Whether that's because of evolutionary limits or weaker selection on populations with higher baselines, we don't know, right? It's just a hypothesis. This is only true for the genetic data. It kind of goes away with the phenotypic data. Interestingly, maybe there's some compensatory plasticity going on there or something. We can move on to body size. So why we're interested in body size? Body size is pretty important, we think, in moderating heat tolerance and cold tolerance in ectotherms. And so we broadly predicted that size would influence, heat tolerance and cold tolerance in general. I'm just going to show the heat tolerance data. But we did think that size divergence would be much less repeatable than the thermal physiology was across our species. And we thought that because there's kind of different expectations based on a few different variables. So first, for aquatic organisms, based on prior studies and based on the oxygen limitation hypothesis, we predicted that there'd be a shift towards smaller sizes because warmer waters, leads to less oxygen, and that could drive the evolution of smaller body sizes. But for terrestrial organisms, it's kind of a mixed bag, right? We have mixed predictions. So fragmentation may lead to greater dispersal, which might select for larger body sizes. Just through plasticity, you might get kind of temperature size rule effects of smaller, urban populations or smaller urban individuals. Or thermal inertia, that larger body sizes might be selected on. So pretty mixed. So going into the results for this, we did see this broad effect, a positive association between divergence in body size and divergence in heat tolerance, right? So the greater divergence leads in body size is associated with greater divergence in heat tolerance. And we broadly saw what we predicted for this aquatic versus terrestrial breakdown too. So you can see in the open circles there, the aquatic studies that we had, those organisms got smaller in the city significantly. For the terrestrial populations, it's a big scatter shot, as you can see. So maybe that's because what we predicted that, body size is just kind of like, grab bag trait that's being selected on indirectly and directly in many different ways. But there's a really strong selection in aquatic habitats due to that oxygen limitation in warmer waters, right? So, kind of like to sum up what I've talked about so far, we have evidence of convergent evolution to this urban warming. There's a lot of variation in how strong it is across, taxa, even within species too. If you looked at that first plot, there's a lot of variation in studies, even that we've done in the same system, right? But you get greater response to larger heat island effects, but there's a big lag, right, with that. And there's a lot of work still left to be done to kind of explain all that variation and to really be able to predict how populations will respond. I'd like to kind of talk about some caveats about this and some future directions as well. So first, caveats, right? We looked at these thermal limits. And maybe that's not the best thing, that's what's out there mostly, but maybe what might predict better how populations will persist or not persist in warming environments would be like their, optimum temperature or other sublethal effects. Or there's just a lot of variation in how people measure critical thermal maximums. And so that might introduce a lot of variation into our results. And then we can think about, well, we looked at the, what was reported as the different environmental temperatures, right? But, maybe this lag that we see isn't as bad as it looks, that they're not keeping up, because obviously, as people have talked about today in the symposium, populations, organisms have ways of buffering themselves from the environmental temperature, right? They can thermoregulate, they can move, they can seek shelter, right? There's Bogart effects that will shield them from this, and maybe that's why they're not responding as much. As we might expect, just, they're not going to track the environment exactly, right? That's kind of unrealistic, perhaps. And if we look across kind of some of the systems that we've looked at in our own labs, in Sarah's lab and my lab, we kind of see this. So if you look at, cabbage white butterflies, we found no evolutionary divergence at all between urban and rural populations. But, these things thermoregulate crazy. They can disperse over long distances. Selection might be fairly weak directly on these, thermal physiology traits. But then if we look at our acorn ants, these ants make their nests just in a tree nut sitting right on the ground. And they have a huge difference between urban and rural environments. So we've seen, mean differences in 4 degrees C within inside their acorns. And so this may drive really strong, selection that we've seen, big divergences like a couple degrees Celsius in their critical thermal maximums, right? And then kind of where to go next, right? So if we're, at least where I think I like to, get more data on, is kind of testing this. Well, how well do these critical thermal minimums and maximums help us predict persistence or adaptation in these warming environments? And one way to do that is to do reciprocal transmit experiments and measure fitness directly, right? And so we've done this in a couple systems. So, isopods and these acorn ants, we've done this both in Cleveland, moving urban and rural populations between urban and rural environments and measuring fitness. And so what you're looking at here in both plots is on the x-axis is the rural environment where it's transplanted to, and the points right in blue are the rural source populations, and in orange are the urban source populations. You can see some patterns here that you can kind of interpret in light of the meta-analysis results. Right, so in general, as other people talked about too, these kind of urban novel environments are pretty harsh. So everyone's doing pretty bad. But you do see evidence of local adaptation for these urban populations within the urban environment. But there's a big lag, right, in different ways, which is interesting. So the isopods, just, that urban populations are really poor, but there's a little bit of local adaptation. In the acorn ants, you kind of get that more classic, crossing reaction norms. But still, like, overall fitness is pretty depressed in those urban environments. And potentially, we haven't directly tested this. Well, we kind of have. But, maybe that's that lag in the physiology that's partly leading to that maladaptation, right? And with that, I think I have time for questions. And I'd like to acknowledge its funding sources and, the people who collected a lot of the data for this as well. Thanks. Yeah.
I might have misunderstood, but do urban heat islands sort of provide us, is the temperature more stable within them, and did you look at if the width of their comfortable zone changes?
Yeah. I mean, we didn't look at variance at all, right? Yeah, so it tends to be like nighttime bias warming, and I think not many people have. Yeah, they do tend. It really depends. People have shown like snow effects. It really depends on your city. So I think it can be really variable from city to city, whether it's less variable or more variable, honestly. Yeah. Oh, yeah. Andrew.
I couldn't remember that bivariate line of temperature and body size.
Yeah.
It seemed to suggest that basically what happened was critical thermal tolerance was shifting in terrestrial things, but not body size. But it was body size, but not critical thermal tolerance in the aquatic.
Yeah. I mean, there's so much scatter in that relationship, but it does there's a pattern in the terrestrial. There's not a significant interaction, so it's statistically no. But yeah, it looks like the kind of aquatic ones are just sitting there, and it looks like the terrestrial ones are responding more. But it's there's no actual interaction there. Yeah.
Time for one more question.
All right. Thanks.
All right, so in order to predict future events, we often need to have an understanding of how things have happened in the past. And this concept could apply to a number of aspects of our day-to-day lives, even things that are notoriously unpredictable. That could include... Okay, that's good.
It's just changing there.
Okay, that's fine. Yeah, that's fine. All right. That could include the weather, sports, traffic patterns, or elections. And the aspect of evolution in the recent past that I'm particularly interested in is local adaptation. And I'm sure you've seen this figure a few times over the last couple of days, these fitness advantages for locally adapted populations, and they're shown as these crossing reaction norms. And one of the best ways to test for local adaptation is through a common garden experiment, which is exactly what I did with the cosmopolitan plant white clover. And I had populations from all across North America and all across Europe, and they were transplanted into 4 common gardens in Mississauga, Ontario, Lafayette, Louisiana, Montpellier, France, and Uppsala in Sweden. But in order to be able to test local adaptation in each of these different environments, we need to have some sort of measure of how different these environments are from each other. So we can do that by determining climatic distance. So I extracted some temperature and precipitation variables for each population of origin and each garden, and they're listed here, and combined those into a 3-axis PCA and plotted them on a 3-dimensional figure that looks like this. And then I can then calculate distances on this figure itself between every population of origin and every common garden as representative of the climatic distance between each of those populations of origin and each common garden. So then I can ask, does the strength of local adaptation differ by common garden location? And strength of local adaptation is going to be represented on a figure like this. So fitness on the y-axis and climatic distance on the x-axis. And if populations are locally adapted, we expect to see greater fitness advantages when populations are transplanted from near climatic distances versus far climatic distances. So the slope on this figure would be representative of the strength of local adaptation. So in this case, common garden A would be exhibiting stronger local adaptation than common garden B. So if I plot my actual data, you can see that Lafayette and Montpellier, so the 2 more southern gardens, or the most southern garden on each continent, is exhibiting stronger local adaptation than the more northern location on each continent, as evidenced by the steeper slopes for the data from those 2 gardens. So does the strength of local adaptation differ by location? Yes, and it appears to be stronger at lower latitudes within a range. Why might this be? Well, there could be maladaptation going on at higher latitudes, or the specific climatic drivers of local adaptation could differ by latitude. And these are things that I explored during my PhD, but I don't have time to talk about today, so feel free to ask me later. Next, I can ask if the strength of local adaptation differs between the native range and the introduced range. So white clover is actually native to Europe, and it was introduced into North America within the last 400 years. So if I remind you of my common garden locations, you might expect to see stronger local adaptation in the European common gardens and weaker local adaptation in the North American common gardens because they've had a less amount of time to locally adapt in that introduced range. But you have to remember that we're also transplanting populations across continent as part of this common garden experiment, so the actual results are a little bit more complex. So in the European common gardens, only populations that were transplanted within Europe are exhibiting local adaptation, whereas populations that we transplanted over from North America are a flat line. And in North America, only populations that were transplanted within North America are exhibiting local adaptation, and populations that we transplanted across from Europe are a flat line. So we see about comparable strength of local adaptation in both continents, but only when populations are transplanted within that range, not across ranges. So we could take this as evidence of rapid local adaptation within the last 400 years post-introduction in North America, but there's also a clear cost of local adaptation. So fitness advantages in one continent don't necessarily translate to the opposite continent. And this could be due to photoperiod, because the sampling range is much higher latitude in Europe than it is in North America. Or it could be due to biotic factors, potentially, and I'm going to talk about one of those now. So white clover is able to produce hydrogen cyanide as a plant defense against herbivores. This requires 2 metabolic components. The AC gene controls the ability to produce cyanogenic glucosides, which are a sugar with a CN group attached. And the LI gene controls the ability to produce linamorase, which is an enzyme that cleaves that CN group and allows toxic hydrogen cyanide to be released. This results in 4 of what we call cyanotypes. 1 that can produce hydrogen cyanide because it has a dominant allele at each of these loci, and 3 that cannot because they don't have both of those metabolic components required. And in natural populations of white clover, there are cyanogenesis declines across latitude. So in very cold environments, as little as 0% of plants are cyanogenic, and in very warm environments, as much as 100% of plants are able to produce hydrogen cyanide. So there's an increase in incline with latitude, and that applies to the individual alleles as well. These clines are also present in the introduced range, but they're a little bit shallower than they would be in the native range, so not quite to 0% in the northern areas. So we can think about a couple of very simple hypotheses from this. At high latitudes, there could be a disadvantage to HCN-producing plants because they're under less herbivore pressure, so they're essentially wasting resources on these metabolic components of HCN if they don't quite need it as much. Whereas at lower latitudes, there's an advantage for HCN-producing plants because they're under greater herbivore pressure, so they're more incentivized to be able to produce HCN. And I could test these hypotheses with another experiment with common garden locations in Ontario and in Louisiana. This experiment also had a precipitation manipulation, which goes along with my original title, but I'm not going to talk about it today, partly because these rainout shelters didn't really work as well as we were hoping. So these results could maybe fit in a future symposium on unpredictability of field experiments, and I'd be happy to organize that if anyone's interested. But we can still ask, does selection on HCN or its metabolic components differ between latitudes? So first in Louisiana, we expect to see fitness advantages for the HCN-producing plants in black. But that's actually not what we see. We see no advantage for any cyanotype in terms of their fitness. And in Ontario, again, not necessarily an advantage for HCN-producing plants, but there is an advantage for dominant AC plants, so ones that can only produce cyanogenic glucosides. So that doesn't really make sense considering the clines that I presented earlier. It gets even weirder when we consider the actual amount of the plant that's being consumed by herbivores. In Louisiana, again, we see no difference across any of the cyanotypes. So it's possible that HCN is not even really an effective defense in this particular environment. And in Ontario, we see, okay, maybe there's effectiveness of HCN in this environment, but there's also reduction of herbivory by only plants that could produce cyanogenic glucosides, but not the HCN defense. So that really doesn't make sense. So is there potential for herbivore deterrence by plants that can only produce the cyanogenic glucosides? So there's no clear evidence of benefits of HCN at either site, but there are benefits potentially for cyanogenic glucosides, but it's only at that higher latitude site. So why might that be? Well, it's actually possible that plants that can only produce cyanogenic glucosides are able to have some sort of defensive effect because some herbivore species contain a beta-glucosidase enzyme in their gut that's analogous to linamrase that could be naturally found in the white clover plant. And when that particular herbivore eats a plant only producing cyanogenic glucosides, HCN could be produced in its gut, and they could be essentially poisoning themselves. So it could be a matter of not necessarily how much herbivore pressure there is in a particular environment, but the actual composition of the herbivore community in that particular environment. So we see stronger local adaptation at lower latitudes. We see evidence of rapid local adaptation post-introduction to North America, and we already also know that there are HCN clines in that introduced range. But we also see stronger evidence of a selective advantage for plants only producing cyanogenic glucosides and only at the higher latitude site, which conflicts with the first 2 points on the slide there. And it could speak to some sort of interplay between biotic and abiotic factors in driving cyanogenesis cline formation and local adaptation in this system, but a lot more work still needs to be done in order to elucidate that. So what does this mean for future adaptation to environmental change in white clover? Well, I don't know if I can necessarily say today, partly because of the complexity of the results and the conflicting nature of the results, but also partly because after dumping all of this conflicting evidence on the white clover system, I decided to do the responsible thing and move on to something else. So I just recently started a postdoc in the Schuth Evolutionary Ecology Lab at NC State, and I'm going to be working on a large-scale field experiment as part of the PERSIST project, which is predicting evolutionary rescue of Mimulus cardinalis. So if you're interested in this type of experiment, Seema and I are both here at the conference, so feel free to talk to us about that, and feel free to talk to us about, or talk to me about my white clover work as well. I'd like to thank a bunch of people that were involved in this project and all of the institutions that were responsible for the Common Gardens as well. I'd also like to thank my co-authors, especially Mark Johnson, my PhD supervisor, and the organizers of the great symposium we've had today, and thank you all for listening.
Okay, everybody, thanks for coming to the last session of the symposium on the unpredictability of evolution. So let's get it rolling. So this is going to be Lars Witting talking about the predictability, predicting the natural selection of life forms from virus to multicellular animals, all over life.
Okay, thank you. I think one of the most essential questions in evolutionary biology, that is if there's any overall direction to natural selection. Now for almost 2 centuries, biologists has assumed that there's not, that natural selection is basically a diversifying process that we can explain everything out from. Now I worked the last 30 years to prove the opposite, that we can actually explain the natural selection of major life forms from virus and all the way up to animals from a quite simple unidirectional selection for an increase in the net energy that organisms use to replicate. And this plot pretty much shows what I will try to explain. It shows mass-specific metabolism as a function of body mass on double logarithmic scale. And the first thing we should notice here is, of course, is that the different life forms, they are segregated by size, more or less. Another thing is if we look at the scaling within the different organisms and life forms, then they differ. In prokaryotes, anything, there's a strong positive correlation between mass-specific metabolism and mass. In protozoa, there's first an increase and then a decline, or maybe an invariance over the whole range. And then in animals, there's of course this negative 1/4 power scaling that most of you probably know. And if you look across the whole spectrum, there's an invariance in relation to mass-specific metabolism. And then on top of this, we should of course not forget the transition from asexual to sexual reproduction and from unicellular to multicellular organisms at the boundary between protozoa and animals. So what I use to explain this is what I call a deterministic selection. That's a forward kind of selection that is based only on the physiological life history and ecological structure that follows from self-replication. What that means is that I do not include a lot of trade-offs and constraints that has evolved by natural selection over time in this model, because those are the things that, if anything, that I should predict instead of put into the models as assumptions. But I do include a few trade-offs that are sort of not all in, like the quality-quantity trade-off and also the different costs of sexual reproduction. And if we take this and look at the sort of the origin of life, then we don't really expect any frequency dependence from interference competition. There might be interference competition, but we don't really expect that organisms are able to differentiate the resource across them. So, and that leaves us basically with a typical R selection kind of scenario. And with this model, it will select basically for molecular-sized self-replicators. So the basic structure of this model is not getting us away from the origin. Except that there's directional selection for an increase in the net energy that organisms use on replication, and that gives a direction to evolution, and that direction, as I'll show, can actually explain the different life forms and also the basically diversity of the life forms across and within. A major component of this directionality, it comes from the unfolding of what I call population dynamic feedback selection of density-dependent interactive competition. And this is an unfolding selection that arises because increased net energy generates increased population growth, higher abundance, more interference, competition between individuals, and eventually that leads to the evolution of a feedback selection. And this feedback selection is selecting body mass as interactive quality, but it's also actually selecting sexual reproduction as interactive quality. And I won't go into the details on that here. You'll have to go to the more specific literature on that. And it selects the body mass in proportion to net energy. So now, from this simple, by including the explicit population dynamics into our model, we get a kind of evolutionary dichotomy between low energy self-replicators at one side, and then on the other side we have high energy organisms that has been selected for large body mass and also sexual reproduction. Now, what this doesn't explain is that it doesn't give you explicit predictions about unicellular organisms like prokaryotes and protozoa. Now, to get to that, we have to partition the net energy into resource handling and the pace or speed of resource handling, with pace being selected as mass-specific metabolism. That puts a direction for selection increase in the way that you handle your resources, but also in the pace in which you're handling the resources. And that primary selection for metabolism is actually selecting mass at the origin of life, because the metabolic pathways they come with a mass in themselves. So originally, the primary selection of metabolism is driving also is carrying along with an indirect selection of mass. And when you take this argument through, you can actually see that the cell of a prokaryote is selected as a metabolic compartment. And this selection is very specific, that it selects against multicellularity because of the quality-quantity trade-off. So if you add an extra cell to the organisms, you're actually halving the reproductive rate. So you have a mechanism here that selects for unicellularity. It's also selecting for incomplete metabolic pathways because of a diminishing return in metabolism as a function of the size of these metabolic pathways. And to get it going, the initial return in metabolism has to be stronger than proportional, and then a proportional function of body mass. If it's not, the selection is going in the other direction. It's going for molecular, non-cellular molecular replicators that use an extrinsic metabolism like virus. So this is what we'll get from a pure kind of R selection scenario with no interactive competition on top of it. But then we have this unfolding of population dynamic feedback selection, and it will select in protozoa-like organisms, it will select extra mass and metabolism. Here the mass is selected partly as the mass of the additional metabolism, but it's also selected partially as interactive quality. And the selection for unicellularity remains in this case. And this selection will continue until the feedback selection is fully developed, and which is also at the point where the metabolic pathways are fully developed, and when there's no return in relation to mass and metabolism anymore. And beyond that point, we have the complete population dynamic feedback selection, and here we have fully evolved metabolism. So the only way to get extra net energy in is by selection in resource handling, and that will select the extra cells as will select the mass that has been selected in that case is selected as extra cells. So there's direct selection for multicellularity when it comes to the complete unfolded feedback selection. So now we actually have four life forms but we want to be sure that the sort of life history correlations within and across these life forms is also following our observations. And in order to get to that, we need to deduce basically the body mass allometries. The first thing I'll focus on in relation to the allometries, that is what I call mass rescaling allometries. These are allometries that reflect situations where the net energy is variation in net energy is selected only from variation in resource handling. There's no variation in the primary selected metabolism. And in this case, when you're selecting a larger organism, then the larger offspring are metabolizing more energy. And in order to select that mass and keep the reproduction stable, you have to make a decline in mass-specific metabolism. And that decline in mass-specific metabolism, that changes the foraging pace of the organisms, and that goes into a selection of the exponent of the allometries by a disturbance of the packing or of the spatial packing of home ranges. Now, this spatial packing of home ranges, that comes from the interference competition that selects for individuals to have very small home ranges, because otherwise you're competing, you're losing energy and time in competition with other individuals. But if you have too small home ranges, then you're actually overexploiting your own resource too much. So there's a balance between these two types of regulation, and that balance, if you quantify that, you get two plots to the left. They show the structural constraints of this packing of home ranges as described as a function of body mass and metabolism and home range. But the lines there come from the predictions, and the dots they are from their mammal data. And when you're instead of having the traits in these equations, insert the allometries for metabolism and home range, you get the two equations below the plots, and these are solved in the plot to the right for a home range exponent of unity, and for a mass-specific exponent of -1/4 when you have a two-dimensional packing of home ranges. So this gives you the allometries when body mass is selected only from resource handling, which is what we expect in larger within taxa in animals. But if you have primary selection of metabolism, then you're basically increasing the exponent for metabolism. Until if you have that on top of this, and we can then go back to sort of the macroevolutionary pattern, where we have the observed pattern below and a predicted one in top. When we go to the prokaryotes, then all the variation in mass is selected from variation in metabolism, and that goes into an allometric relationship that is close to linear. It is predicted to be 3/4 or 5/6, depending on the dimensionality. And then when you go to protozoa, they start out at prokaryotes, and then they go more and more to selection of mass from resource handling. So they go to what you see in animals, which is the -1/4 power exponent, and the invariance you see here between the predicted life forms of animals, that arises that when you predict when you select a new life form, and you become better at extrapolating resources from the environment, that is likely to be a very slow process, because you have to restructure the whole phenotype. And if it's sufficiently slow, then the metabolism that is lost during mass rescaling selection of this new life form, that leaves sufficient time for the primary selection of metabolism to back select that metabolism that is lost, and you get the invariance. And then, of course, we also have the prediction between asexual and sexual reproduction, and between unicellularity and multicellularity. And when on top of this you add speciation and interactions between species, then you also get a mechanism for maintaining the sort of the diversity of organisms, so that they are not just all piled up at the end point of evolution that is predicted from this. And with this, I think I will stop and thank you all for your attention. And all the original work with all the details, you can find that at my homepage up there. So, thank you.
So, hi everyone. During my PhD, I had the opportunity to work very broadly on predicting evolution. And that's actually what I'm going to be presenting to you today. So first, I'll start off with sort of a theoretical background, both into the history and the present-day challenges of predicting evolution. And why am I giving you a bit of history? Is because predicting evolution, I think, is an old field. And it motivated, so old that it even motivated the modern synthesis. And the present-day challenges that we're faced with in predicting evolution are actually inherited from the conceptual framework of the modern synthesis. And then if that theory was like a bit rough, I'm going to make it a bit more concrete and give you an empirical example where we worked on antibiotic resistance in E. coli, looking at different types of interactions. And here is a bit where the ethics comes in, because in a health and biomedical context, controlling pathogen organismal evolution and the evolution of particular traits like antibiotic resistance, controlling these things is really interesting to us. And so I return to the history, but in an ethical context. And then I ask, is this a present-day challenge? Should we be thinking about controlling evolution and the ethics of it? And so let's start off with the theory. This is a very, very brief history. So I think people have been thinking about predicting evolution as soon as we had domestication of plants and animals, because people were thinking, is there a limit to trait evolution by artificial selection? And that's in fact where the debate begins, as far as I can tell, from the literature. And this is between Malthus and Darwin. And Darwin thought there was no limit to trait evolution by artificial selection. And he spent a good deal of time documenting all kinds of examples to support the idea that all of life's diversity came about through processes similar to artificial selection. Then fast forward a little bit, his cousin, who was at the time interested in predicting weather patterns, Galton, he reads The Origin of Species and he's like, wow, let's do this. This is cool. And he begins the biometric school. The biometrists were interested in understanding the quantitative genetic basis of traits. Remember, this is before Mendel. So predicting evolution or heritability, all of this was very jumbled up. And the questions that they were asking were can we predict the traits of the F1s or the children or the grandchildren and beyond by regressing them on the parentals? That's why Pearson is here. This is regression. So then the next thing that happens is the modern synthesis. And when I say the modern synthesis, I mean the joining of Mendelian genetics with biometry to create the field of population genetics. That's why I've got these two big guys, Fisher and Wright, who I'm sure you're familiar with. So they were working on managing animal populations. And both of them came up with their idea of fitness landscapes, which I'll talk to you in a moment. And then the final contribution sort of intellectually comes from Stephen Jay Gould. I won't mention this, but I think it's really important in how we think of predicting evolution, because he gave us the idea of historical contingency and phylogenetic thinking. And so if I haven't convinced you that the biometricians are not, they were doing something else, they weren't doing predicting evolution, here's this quote from 1893 by Raphael Weldon. So he says, the problem of animal evolution is essentially a statistical problem. To estimate how populations are changing, we have to measure, and then you have to look up the quote. There's a long Victorian-style list of things, and I summarize it as the frequency, fitness, and heritability of traits. Maybe that's not, please look it up, and let me know if you disagree. Then he goes on to say that these are questions of arithmetic that will allow us to know the direction and the rate of change in these species in the present day and to speculate about their past and their future fate. To me, this really sounds like a research plan in predicting evolution. And this is one of the fathers of biometry. So yeah, let me give you now a bit of the modern theory and the fitness landscapes and a little bit of the flavor of what is fitness landscapes. So it's a powerful toolbox for predicting evolution because fitness landscapes are a map of where evolution is headed. Here I'm showing you a diagram that's depicting Fisher's geometric model, which has a single peak. We have two phenotypic axes, and the heat map is fitness. And in this case, evolution is fairly predictable. There's only one peak, and so mutations have to be in the direction of the peak in order to be adaptive. And sometimes the Fisher's geometric model fits empirical data pretty well, and sometimes it doesn't. And in that case, we have a bit more complex models. So this is just slightly more complex. We have 2 peaks here, and this is similar to the types of models that Wright was thinking about. And here I said there's 2 peaks, and that means that the landscape is a bit more rough. So it's more difficult to predict where evolution is headed. I have the same mutations I showed you before, but here they're both adaptive because they're both in a direction of a peak. But an interesting thing happens with a multi-peaked landscape, which is when you combine those 2 adaptive mutations together to get the double mutant, you observe epistasis. So in this case, it's deleterious. Epistasis is annoying, but it gives you an estimate of the roughness of the fitness landscape. So it's also informative about something underneath that's happening. And if you were paying close attention, you probably noticed that I glossed over another type of interaction, which is gene by environment interactions. And so I'm showing it here by overlaying these 2 ideas of the single peak and double peak landscape. In this case, the purple mutation is easy to predict because it has no genotype by environment interaction. And the red one, it can be positive or it can be negative depending on the environment. And so I think, I hope I've convinced you that evolution is less predictable when there's strong interactions, but also that fitness landscape models are useful because they help us make sense of unpredictable interactions. Epistasis gives us an idea of the roughness, and G by E tells us how fitness landscapes change between different environments. And so to kind of drive that home, let me give you an overview of the empirical work that we did. So in my PhD, we were really interested in understanding how do fitness landscapes change between different environments. And we were interested in ecologically relevant environments because that's the good stuff, right? So we thought that bacteria might encounter heat and antibiotic together in a patient that had a fever and had been prescribed antibiotics. And we wanted to know how this landscape looks like for the bacteria as it's evolving. We were interested in random combinations of mutations. And we looked at the category of mutations that we know from experimental evolution is most often used for adaptation, which is knockout mutations. So we picked some knockouts that are naturally segregating in wild E. coli populations, but that don't interact with the other gene that I have, that's RNA polymerase B. So this is the target of rifampicin, and we picked 3 SNPs that confer different degrees of antibiotic resistance. I made all 24 possible combinations of genotypes, and then I screened them for their fitness in the 12 different environments. So I used a gradient of rifampicin antibiotic and a gradient of high temperature. This fully factorial design allowed us to look at interactions, because that's what we wanted to know. How does epistasis change between environments? And so, I mean, I did it was a bunch of work, but here's the summary. I'm showing you just one metric of epistasis. I have a bunch of other metrics, and they all mostly agree. This is the roughness to slope ratio that you see on the y-axis, and the x-axis is the antibiotic concentration. So what you're seeing, this trend line, is that as antibiotic concentration goes up, epistasis goes down. And if you remember from earlier, I told you that the roughness of the landscape is inversely correlated with the predictability of evolution. So the trend line shows a decreasing trend of epistasis. That means that the predictability is getting like more predictable as we add antibiotics. So the landscape becomes smoother as you add antibiotics. We didn't see any effect of temperature, and this could suggest that fitness landscapes remain relatively unchanged for smaller environmental perturbations that organisms are already adapted to. But I mean, please go out and check my hypothesis. Is evolution more predictable in adverse environments and less predictable in benign ones? Maybe that's happening. Okay, so the last part of the talk, why do we want to predict evolution? What are evolutionary predictions good for? I think in the symposium today, we've heard a lot about testing our assumptions about how evolution works. And through evolutionary experiments, natural experiments, lab experiments. But in health and biomedicine, there's a lot of interest in predicting evolution for the purposes of control and just even preparation. So if we could predict the flu strains of next year, we could make great vaccines. If we could predict how organisms evolve to become resistant, we can make better combinations to prevent them from becoming resistant. And in fact, both of these are uses that we already have for evolutionary predictions. We're already doing this. And this sort of brings me to the fact that actually the idea of controlling evolution is not new. It's as old as the field itself. So the term eugenics was coined in 1883 to refer to controlling human evolution. And as you can see from the graph, the phrase eugenics was used about as often in the evolution literature as predicting evolution is used today. This is not like a trendy field. It's as old as the modern synthesis. So for the rest of this talk, I'll follow the convention of historians. So when I say eugenics, I'm really using it as a precise jargon to refer to a self-identified historical political movement. The main goal of that movement was to use scientific theories of heredity and the state to improve human phenotypes, at least to breed out genetic diseases and sometimes even select for superhuman traits. Eugenic political movements had horrific consequences for marginalized groups worldwide, and many evolutionary biologists supported them early on, but most withdrew their support by the 1930s. Not all the scientists who helped shape our understanding of predicting evolution supported eugenics, and in purple, I've highlighted the ones that were opposed to eugenics. It's uncomfortable to acknowledge that a line of current scientific research shares its origin with the horrific state project of eugenics. But we need to acknowledge that history to make sense of the potential dangers inherent in the possible applications of predicting evolution. So, sideline of this, I also noticed that there's like a trend. So I think that some of the eugenic the most critic people were trained as biologists, and like the ones that are most proponents might have been more trained as like physicists and statisticians. I don't know, that's just sort of like a thing, like neat. Anyway, back to the drama. So the goal of controlling evolution holds a moral dilemma. It alienates the target population from its own evolutionary process and puts the controller in the driver's seat. That's why evolutionary predictions are not used today to control human evolution, because that would impinge on the reproductive autonomy of humans. Eugenicists understood this moral dilemma, but in a different way. And you can see that in the logo on the right with the slogan, "Eugenics is the self-direction of human evolution." In other words, humans are put in the driver's seat of their own evolutionary process. The optimism of the slogan highlights that eugenicists held a different moral lens than our own. In ethics, this is called axiology, and it's the moral lens that lets us decide like what is the target population and how control will be implemented. This is like not biology that I'm talking to you about. This is like ethics now. This is interdisciplinary work. And so my point basically is that like we need to understand our own history so that we can better interface with other people who are subject experts as well as policymakers and perhaps the general public. Because if we don't understand our own work, then we can't do the interdisciplinary work that has to be done. So in summary, I've shown you that evolutionary predictions can and already do make our lives better. But in most cases, interactions between genes and environment make predictions rather difficult. My experiment found that evolution might be more predictable in adverse environments as compared to benign ones. And after over 100 years of research, we might be closer than ever to controlling evolution. Therefore, it's our duty to acknowledge and communicate the moral issues inherent in this field. So thanks, everyone, all these my PhD supervisors and everyone who supported me. And if you want to learn more about my work, you can watch it in the Hamilton Symposium video. Thank you so much. If anyone has questions.
Don't you think it's maybe misleading to say that Darwin was opposed to
should be working.
So I think the good distinction would be to see if you are opposed to positive or negative eugenics, because Müller was for eugenics, Huxley was for maybe everybody. So I'm not sure if it's a good line.
I think Darwin is really tough.
Yeah, I think the good distinction would be the relation to positive or negative eugenics and how it mimics natural selection
Good thing you're waiting.
clarifying or positive directional selection. Maybe we can talk.
Okay, let's get going with the next talk. Try and stay on time. Okay, so now Joe Travis is going to tell us all about identifying the locus for predicting evolution.
Thank you, Brian. I begin with this illustration of the pirate perch, Aphrodotyrus cyanus. This is a fish whose distinguishing feature is that its anus is underneath its throat rather than at the back. Now, I defy anyone in the room to tell me that this was a readily predictable outcome of evolution. Okay, we agree. Having established that evolution can be unpredictable, I want to turn to a more focused discussion of these issues. First, I will use published work to describe a study testing or a theoretical prediction about evolution in the guppy. Then we'll criticize this work and ask whether it really actually illuminates the issues we've been talking about all day. And finally, I will respond to that criticism. So you can think of this as thesis, antithesis, and synthesis. So let's begin. This is the Trinidadian guppy. It occurs throughout the island of Trinidad. The story I want to tell you occurs in the rivers of the Northern Range Mountains. At the low elevations on the left, these rivers are wide, the water moves slowly, and it seems nice, except that guppies live with a high diversity of piscine predators. Their lives are nasty, brutish, and short. As you go up in elevation, the rivers narrow, the water runs faster, the piscine predators drop out one by one. By the time you're about 200, 250 meters elevation on the right, the streams, at least in the dry season, are a series of pools and riffles, and guppies live largely in the absence of predation risk. The low predation populations, this is the continuum. I want to emphasize that, but I'm going to contrast the extremes, the high predation, low predation. But I emphasize it is a continuum. The low predation populations upstream are descended from the high predation populations downstream. A number of papers have shown this. This is an illustration from a recent paper by Jim Witting and colleagues. Now, there are many traits that differ between guppies upstream and downstream, or low and high predation. This slide illustrates two of them. You can see, without the benefit of any generalized linear mixed model, that the fish on the right, the males, are larger and brighter than the fish on the left. So that tells us two traits that are different. It implicitly tells us about a third trait. And to see that, you have to know that males don't grow after they mature. So the size you catch them is the size at which they matured. And larger maturation is associated with a longer time to maturation. So this slide also shows you that at low predation populations, they delay maturity. Can this be explained by the alleviation of predation pressure? You'd think so, because the slide says so, right? Not so fast. Because a gradient in predation pressure can create a gradient in other ecological agents of selection. This is a typical ecological graph with lower predation pressure, higher population density of the prey. Density is a surrogate, or a proxy, if you will, for agents of selection like reduced per capita resources, greater interference competition. Now, when you have a situation like this, whether theory predicts a delay in maturity depends on whether and how each agent of selection acts on the demography of the prey. This is well known in theory for many, many years. I show you papers on the left by Abrams and Rowe and Anna Gardmark from 1996 and 2003. Empirical work is finally catching up with this. In other words, to test the theoretical prediction about whether the forces on this gradient predict a delay in maturity, we have to know which force is it. Is it predation or is it density? To simplify this, how will we know? Well, we can distinguish the effects of predation from density by following new low predation populations as they establish and grow. The left-hand panel shows you population numbers over time. goes up and plateaus. The solid line is the expected dynamics if predation drives selection. The dashed line, what you expect to see if density drives selection. Notice that they are exactly the same. There are two dashed lines showing some difference in initial density, so you can't distinguish these from population dynamics. However, on the right-hand side, the selection gradient for delayed maturity ought to be, if it's predation, ought to be very great right at the beginning when density is low and they've been released from predation. It will decrease over time as the populations approach their adaptive peak. On the other hand, if it's density, then the selection gradient initially will be very low, not high. As in the predation case, it will increase as density increases and then decrease as the populations approach their adaptive peak. So the question is, or the point is, by distinguishing the effects of predation from density via the dynamics of age at maturity, we can know which specific predictions to test. How are we going to do this? Well, we can transplant high predation guppy stock upstream into 4 reaches without guppies and recreate the natural establishment of low predation populations in a controlled experimental design. In other words, let's experimentally recreate nature. When we do that, this is the trajectory of population dynamics in our four populations. I want you to notice the word intact and thinned. 2 populations have intact canopies. The other 2 have thinned canopies. That will become important in around 8 minutes. Check your watches and pay attention. Notice that by about month 30, population growth occurs to about month 30, and then they enter a period of fluctuations. So there's an initial period of growth, and after about month 30 to 35, things begin to stabilize. Annual common garden experiments on the F2 fish show evolutionary change begins after month 30. I want you to look, please, at the top panel, which is the age at maturity. This shows you F2 fish from common gardens compared to the high predation control, which is the horizontal line in those graphs. Notice that the first 2 on the left, upper Langerhans and lower Langerhans, don't change until 3 years. 1, 2, 2011, 3 years after introduction. And then they continue to change. On the right, they don't start to change until 2 years after the introduction. This suggests that density is the driving force, not predation. Well, okay, fine. When does density-dependent selection theory predict the evolution of delayed maturity? So I will verbally summarize whole books of theory by Brian Charlesworth. First, when the rate of population growth is consistently coupled with population density, and second, when the effect of population density on population growth rate acts consistently through recruitment, which is some combination of adult fecundity and juvenile survival. Well, we have these populations. We followed them for years and years and years. What do the data tell us? Patterns in the monthly change in log density are fit better by a density-dependent model than a random walk. These are state space models of the monthly change in density. Decreases tend to follow increases and vice versa, a signature of density dependence. Condition 1 is fulfilled. Condition 2 is about recruitment. And here you see the raw data for each of our 4 populations. Recruitment decreases as population density increases. You don't see this for adult survival. So the conditions under which theory predicts a delay in maturity are met. So we have managed to predict evolution in the Trinidadian guppy using mathematical theory and a controlled experiment. I will now criticize this work. Of course evolution is predictable. The guppy study standardized the genetic, you could tell a reviewer said this, right? The guppy study standardized genetic background, initial conditions, and the ecological milieu of selection. Exactly. My response: the experimental constraints that allow evolution to be predicted successfully actually allow the loci of unpredictability to be identified. And then we can start asking questions about why evolution and when it is unpredictable. And I would suggest 3 possibilities: genetic background, what genetic variation is available when populations encounter a novel selection pressure; initial conditions, where are they when this starts; and finally, variable ecological milieu. That is to say, when a novel selection pressure emerges, through which contingencies does it act? I'll give you examples of each of those, or things that might fit into each of those examples. This is the sailfin molly male, Picilia latipina. Isn't that a beautiful dorsal fin, right? Come on, admit it. It's a beautiful dorsal fin. Come on, help me. Agree with me. In some genera of peciolated fish, males have exaggerated ornaments. This is what sexual selection theory predicts, and species in which males court females, and the major action of sexual selection is through female choice. This happens repeatedly in the peciolid family. Here I show you a phylogenetic map of the peciolids, and I want you to see that different traits are co-opted. Different genera co-opt different traits as exaggerated male ornaments. The caudal fin elongation in swordtails is different from the dorsal fin elongation in mollies, is different from the color accentuation in guppies. So this could be simply a function of what genetic variation was available to respond to selection. It also reminds us a little bit about what we're trying to predict. Sexual selection theory predicts exaggerated male ornaments. It is agnostic as to which traits are co-opted to make those ornaments. My second example is from a model of conflicting selection pressures, and this is by Don DeAngelis and colleagues, and it's a model of gastropod predation on mollusks based on a lot of data they accumulated. And what I want to show you, if you look at the upper left-hand panel, all four panels show you the age at which reproduction begins moving increasing left to right, and the amount of energy invested in the shell increasing top to bottom. In the absence of predation, the adaptive peak is let's see if I can make this little beam go without blinding anyone. The adaptive peak is here with very little investment in the defensive structure. As predation increases, initially the adaptive peak stays there, but with a greater increase in predation, a second adaptive peak emerges, in which you should invest a good bit of energy in hardening the shell and reproduce quickly. With even more predation, a third adaptive peak emerges. And the point I want to make is that if you have populations in different places on this landscape initially when predation commences, then they could be in different basins of attraction and end up in different places, and evolution looks very unpredictable, and it has been, even though the game rules are the same for all those populations. So, and the third example goes back to our focal streams, and now you have to remember what I told you 8 to 9 minutes ago, that 2 of those populations had open canopies, 2 had intact canopies. Color patterns evolved in males in experimental populations in response to alleviating the direct effects of predators. These are F2 common garden males. The high predation control is the top row, Guanapo. The 4 experimental populations are the other rows, and these are illustrations of what males look like under different lighting conditions to highlight green inflorescence, blue inflorescence on your left, orange spots and black spots on your right. If you actually look, and this is from paper by Daryl Kemp, some male colors evolve similarly, that is the orange spots, and others differently. And here's orange spot cover. Here's our control, Guanapo there, our 4 experimental populations. Notice that orange spots decreased in all 4 populations, but notice that in 2 of them, black spots increased and the other 2 black spots decreased. And it's easy to say, see, evolution's unpredictable. But this is my example of a contingency. Because if you then look at the ambient light levels, if you look at black spots on the upper left, you see the greater the light level, the reduced level of black spots. Green inflorescence shows you the same thing. So, I've told you about predicting evolution in the Trinidadian guppy. I've criticized this work as an overly constrained experiment, but I offer to you the thesis or the synthesis that by having experimental constraints, we actually allow the loci of unpredictability to be identified and find some interesting questions to ask. And some of this reflects what you've heard earlier. Andrew talked earlier this morning about the more you learn, the better your prediction can be. The more you learn about lighting environments, the better your prediction of colors will be. So that's the argument, and I'd be happy to answer any questions in the time available. Or not, given that was the chime. was that the chime or the bell, Brian?
You got 30 seconds. Well, 3 seconds. Now, I guess we should roll on.
For whom the bell tolls.
Okay, all right. So let's get rolling. We'll wrap up this symposium now with the last talk of the day, which I will give. So first off, get going. Let's just say what great set of talks today, right? So lots of really cool talks about predictability of evolution. Thanks to everybody giving talks in this symposium. Lots of great work. Really, yeah, loved it. Really cool. Really diverse perspectives. I hope everybody's noticing that. So now it's my job to talk about this field by taking a step back and sort of trying to take a look at an assessment of this field. Where are we, right? What's the current state? What are some possible routes forward? Spur some discussion. And just be forewarned right now, we'll be raising more questions than we'll be giving answers in this talk, but hopefully it's going to make you think. No real data from empirical stuff in this, of empirical evolutionary prediction here, other than empirical data from you guys answering surveys. Okay, so let's get started with this one big question. So what is the science of evolutionary prediction? The symposium is supposed to be really trying to advance the understanding of this field. So is this a unified, coherent field, or is this like some kind of ragtag bunch of researchers with a common thread sort of running through our research programs? All right, so maybe it's somewhere in between these two things right now, but I do think this field exists. It can be defined like this. It's basically the field of inquiry into the predictability of evolution from both a fundamental perspective and an applied perspective. So some of us are really focused on trying to figure out how the heck this universe works. We want to figure out how the evolution process works and understand and gain better understanding. Some of us really want to just get on with the predicting and solve practical problems. And some of us, straddle both. But I think this does encapsulate the general motivation and what it is that is a field of inquiry into the science of evolutionary prediction. And if you take a look at this, it sounds a lot like meteorology, actually. And that's a pretty coherent, unified field, so maybe this thing can be too, right? So let's get started on what I'm going to talk about for the rest of the talk here. We're going to do 4 main things. We're going to talk about the diversity in this field, what it is that we really want to do as a field, what we need to do, and how we might actually get there, maybe. Okay? So first off, this is one diverse field, which hopefully everybody could have already seen from the talks today, but this is a large tent. Lots of different perspectives are brought to bear on trying to understand something about understanding the predictability of evolution. Just look to today's symposium. Check out tomorrow's symposium. There's a plug for that. Other talks at this conference, right? But don't just take my word for it. So we just recently did a survey to some of the attendees here at this conference, and if you filled out the survey, thank you, all right? So Marius and I did this survey the last couple of weeks, and if you saw his talk this morning, you already heard a little bit about it, but the survey is a slice representation of us, right? Researchers who are interested in this field. So what the heck do we think is going on in this field? Who are we? Where do we think we're going? So I'm going to be referring back to this survey a little bit later as well in this talk. But it really does show, just this little slice, how diverse we really are, right? So when we ask this question, what really describes you best? There's 14 different answers that people identified, right? Including philosophers and physicists, and just this one little survey right here, right? So even if you just took a look at the 6 most common or most represented in the survey, these are pretty diverse perspectives being brought to bear on this topic. And one other question we asked in the survey really drives home this diversity and why this diversity really matters to try to make sense of this field. And that's when we asked something that actually Andrew Hendry was talking a bit about earlier today. What does prediction mean, really? So this question was, what does predicting evolution, or the predictability of evolution mean to you? And wow, a lot of different things, it turns out, right? So the answers were really diverse, very thoughtful, really cool. But it really points out that just in this slice of folks who are interested in this field, people are thinking about this very differently. Different meanings of the word predict, different things they want to predict, different time scales, different precisions they're talking about. They clearly have different motivations that go into their descriptions here, different ways of predicting. This is diverse, right? Like I've been saying. But you might look at this and say, what a mess, right? We're just in a swamp, and how do we find our way out? We're just going to stir up the water rather than trying to propel ourselves forward. But actually in here, I think, out of all these things right here, it's pointing to themes, right? Things that can help us make a framework. And so we're going to try to work toward a framework maybe to help make sense of this field, to frame the problem and help guide us forward. So that's what we're going to talk about here. We'll go through these ideas that we just talked about there. So what we want to do is predict evolution, right? We want to figure out how to predict evolution and how predictable evolution is given some kind of understanding. But to do this, we really do need to first know, what does prediction mean? Okay. Now, Andrew Hendry talked about this earlier today a little bit, and you can check out his paper from 2023, when it was published, right? Just last year. And rather than talk about all those different things, I think that pretty much everything that we talk about when we use the word prediction in evolutionary biology sort of boils down to 4 primary types of things that we're talking about when we use the word predict. And I think one of them really is the main source of prediction that we really focus on in this field, but all of them are important. But there's really one type of prediction that's really prediction. Right? So here's what I mean. And understanding these, I think, becomes important to understand how we're taking these different approaches toward this question and how we might go forward. So sometimes we use the word prediction, and really what we mean is explanation. Right? And so what we're really trying to say is this is how something works. This is how something functions. Something predicts something because there's a causal interaction, right? And that's really not a prediction itself. It can really help us understand something to make predictions, but it's not an actual prediction, right? So we should recognize that to have a common terminology. Sometimes we literally mean prediction. That's good, right? So this is where we've got some kind of data that yields an evolutionary outcome. This implies some kind of model, causal model, correlational model. There's something in here that's mapping on something that we think will take data, some kind of set of conditions of life and its surroundings, to an evolutionary outcome. We can also try to understand this in reverse, but that's a prediction, right? Prediction. Another way that we use this term refers to something very related and very similar, but not quite a prediction, which is repeatability. Right? Repeatability is where you have the data and you can compare it, and you'll see evolutionary parallelism, evolutionary convergence, right? Those are repeatable patterns of evolution, right? So that may say, wait a minute, well, that tells us that evolution is predictable, right? Yeah, it tells us if all we see is that the patterns are repeating, evolution is predictable, but who's doing the prediction? The predicting. It's not us, it's nature is doing the predicting. So if nature sees these sets of conditions and can produce the same thing over and over, that's great. But for the science of evolutionary prediction, this prediction really is referring to us making the predictions, right? So repeatability is, maybe it's a weak form of prediction, but it's not quite there, and it feeds into prediction, all right? So slightly different. And then one other thing that we often mean by this is determinism, right? It's something slightly different, and determinism isn't really prediction, but it is related because it's talking about deterministic forces which can lead to repeatable patterns, right? Like deterministic force, natural selection can lead to repeatability, which then feeds into prediction, right? But they're not really prediction, right? So in the field, I would say these are the 2 most commonly used things. And this is the one that I believe, in my opinion, is the real one that was like maybe a gold standard or something, but it's the main thing we mean actually explicitly prediction per se, right? But it's good to be able to have a common terminology to talk with one another. So once we know what prediction is, what do we want to predict? Okay, so we want to predict different kinds of things. And I think that this is important to recognize that there's different dimensions of what it is we want to predict. And these dimensions might help us wrap our mind around what we're trying to predict, how we go about doing it, way forward. So I think the main dimensions of variation in what it is we want to predict are these prediction scales. Right? Most of us want to predict something that varies along a biological scale, a time scale, and a precision scale. Right? So we want to predict something from, thinking about the genes, the phenotypes, the fitness, or even higher in the biological hierarchy. We want to predict short-term, long-term evolution, and different kinds of precisions, like super precise, very specific nucleotide substitutions, or just it's going to change, I think, or it won't change. Right? It can be very broad, very precise. But once we just take a look at this, we might already see, I wonder where the studies that are being conducted today often fall. Maybe it's going to be a lot easier to make accurate predictions here than here based on what we know now. Or maybe, where is it that we're going to need a lot of information to make a good, accurate prediction? That's when our studies are like down here, whatever, right? But I think these are the major ways that what it is we want to predict can vary. So what is it that we need to do? What we need, I think, is a roadmap forward, right, to know what should we be doing in the short term, what should we be planning to do in the long term, right? To do this, I think we need some kind of common conceptual framework to start with. And I don't know that it exists yet, but maybe one way to start is to just start with something like I was just describing and start adding on things like this, right? So these are I think maybe there's one more dimension, but these are the main ways I think that studies tackle this problem. They have different dimensions of the prediction scale, and then they have different types of their prediction, or the source of their prediction. Right? They're coming up with some way that they came up with the prediction in the first place. Correlational model, it could be AI, right? It could be a causal model, a really simple causal model, which is where I think a lot of the talks in the symposium came from. Could be, really complicated theory, could be new, really complex networks, whatever it is, right? But then you start placing these studies that exist in here and start pinpointing gaps maybe, like, oh, no one's really working here. Why is that? Where do we need to be studying in order to understand X or understand Y? Well, here's the problem. The problem is that we need to solve this. To solve this, I have to be able to predict this kind of change. How am I going to do it? Well, maybe this is going to help point you to, well, you're a creek without a paddle, right? Or, oh, that's going to be simple. You should do this, right? So maybe starting somewhere like this might help. So if we get somewhere like that in the near future, then we can start asking ourselves, how the heck are we going to get somewhere where we really are advancing the field once we have something maybe in place? But now I'm going to return back to this survey I was talking about earlier. So we asked a number of questions that we thought were useful in a forward-looking type of set of questions for the survey to see what does the field think about where we're going. What should we be doing now? To see if there was any consensus or whatever. So the first question is basically, what's the most promising approach to advance this field? What should we be doing right now? What kind of studies should we be conducting? And everybody said the same exact thing. We all know what to do, which is really cool. No, they didn't. So what they said was a lot of different stuff. Everybody has different ideas, right? Now we should do this. Now we should do that. Right? And maybe we're right. Maybe all these things are great. And the only way to tackle this problem well is to take a really big approach. Because like if you got a bunch of darts and they all look good, throw them all at the target, right? Maybe one's going to stick, right? And so maybe this is the way forward, or maybe it's because we just don't know yet and we need to get together and do better synthesis and figure out the best way forward. Another question we asked was, how long are we going to have to wait until we actually have peaked in our understanding? When are we going to really know how to predict evolution to the best of our ability? And surprising to me, most of you guys think that it's not going to take that long. Right? It's only 5 to 30 years. This is only like 30 to 100 years. So within 100 years, we're going to be the best ever. That either means it's right around the corner, man. We're going to be really good at this in a hurry. We're not going to have to wait that long. Or we're never going to be very good, right? We're almost there. I think we're here, right? Like we're way far away is what I think. But we clearly don't have a consensus view on that. The last couple questions we asked real quick is this ever going to be really very effective in a bunch of societal applications? And we definitely think it will. I mean, it already is having some impact, but there's a diversity of opinion here, but vast majority not only think that, but actually we're really pinpointing the same kinds of themes here of where this is going to have the most success, which was in areas of those regions right there. And then the last question I'll talk about is this question of, will our impacts on these societal applications have a net positive or net negative impact on the planet and society, right? Meaning, are we going to make the world better or not? Maybe not too surprising is that nobody said it's going to make it worse. Nobody wants to be studying a field, I think, that's going to make the world a worse place, I guess. But so most people do think it's going to have a good positive effect and make the world better. Hopefully that will turn out to be true. Some of us are like, I don't know about that right now. But that's sort of the state of the field. So to conclude real quick, there's 2 major motivators to doing this kind of work. There's 3 major predictive scales, I think, and there's 2 major types of predictions. So that helps frame the problem. What we know right now is that some evolution is definitely predictable. We also know plenty of evolution does not appear predictable from what we're doing right now. And some may never be predictable. If there is true inherent limits like from chance, we don't know that. The field from our survey, Mario's talked about this morning, does not have a consensus on that. We're not really sure about where we're going, but there's some patterns. And we have marches and orders. Now we don't. So I don't really know where to go yet, but we need to discuss this as a field to help advance it. But that's where I'll end it. So thanks for attending. Thanks for speaking in the symposium. That's it. That's all I got.